Dear friends, today we are going to discuss about the histamine as well as the histamine antagonist. First, we'll go through the histamine. See, the histamine, when the chemical structure of the histamine, when we will be right, it is in the form of imidazole. At the fourth position, it is having the ethylamine group. If we give the numbering for this one, always we are going to start with the heteroatom, as we know. Next heteroatom should have to get the lowest possible number. Then this is the fourth position, this is the fifth position. Here when comes to the numbering for this one, this is the first, this is the second position. When we will write the chemical name for this one, at the fourth position, at the second position of the side chain, it is having the amino, that is 2 amino, ethyl. This all present at the fourth position of the imidazole. For 2 amino ethyl, then it is called as the imidazole. This is the chemical name for this. Then next we have to become to know where it is present exactly. Histamine, it is present in the mast cells. Histamine, it is in the form of protein complex, okay, which is present in the mast cells, okay, in the basophils, and as well as in the GIT. Especially GIT means it is present in the parietal cells, we know, which is responsible for the acid secretion, and as well as it is present in the hypothalamus. Histamine present in the GIT, mast cells of the GIT, present in the basophils, present in the hypothalamus, that is the posterior part of the hypothalamus, in the CNS, that is central nervous system. Where it is present, we came to know histamine. Now, how it is going to be synthesized? Histamine, it is synthesized from the histidine. For that, you should know the structure of the histidine. Same at the fourth position. When you see the histamine structure, the extra thing is the carboxylic acid which is present. This is called as the histidine. Here, especially in the mast cells, one enzyme is available. That enzyme, it is called as the histidine decarboxylase. Remember the enzyme histidine decarboxylase, which is going to be remove the carbon dioxide. It is going to be removed from here. Removal of the carbon dioxide, it is going to be produce the histamine. This is the synthesis of the histamine. Okay, histidine undergoes the decarboxylation, especially in the mast cells. Then once it will be undergoes the decarboxylation, removal of the carbon dioxide will be takes place. Then it will be converted to the histamine. This histamine, it is responsible for the many things. For example, you take for the allergic reaction, histamine is responsible. Acid secretion, histamine is responsible. Any inflammation related things, histamine is responsible. But this histamine, it is producing its action through the 4 receptors. Histamine producing its action through the 4 receptors. What are those 4 receptors? We know, we studied that in the pharmacology. One is called as the H1, another one is called as the H2, another one is called as the H3 and H4. This histamine produces its action through these 4 receptors. Is it we required these actions through the H1, H2, H3, H4? No, we don't require. We have to be control this histamine. We have to control this histamine. Okay, before discussing how to control, how to inhibit the histamine, we will go for the its mechanism of action. Histamine, how it is going to be released, and where it is present, where it is located, okay, and what type of action it is going to be produced. First we study about that. Now this is the mechanism of mechanism of action of the histamine. How the histamine, it is going to be produces in our body. Okay, this process is called as the exocytosis. Okay, where histamine it is going to be released. Just see in this case what happened. These are the mast cells. Already I told where they are present, all the organs. Okay, many of the body organs it is present, like basophils and different things, hypothalamus and GIT in the parietal cells. Then at that time what happened means immune and non-immune allergens. Okay, this I given the shape for the remembering purpose. They are not in this shape, but just for explaining the mechanism of action. These allergens, they are in the different forms, like a dust, like a pollens, like antigens, like antibodies, like xenobiotics. Okay, that is the foreign body materials are different forms. They are present in the food, or they are going to be take through the water and different things through the different way. It will be releases in our body. All they are causing the release of the histamine. How this immune and non-immune allergens it is binds to the antibodies. There are number of antibodies, they are present in the mast cells only. They are going to be bind with this mast IgE antibodies. Here you might be see. This is the allergen. Okay. These are the antibodies. Allergens bind with the antibodies, and what it will be going to be do in this case means allergen binds with antibody. Then they are going to be release the histamine from the mast cells. Okay. Then releasing of the histamine from the mast cells, this process is called as the exocytosis. What it is called? This process is called as the exocytosis. Okay. Then by this process they are going to be release the histamine. Okay, not only it is going to be release the histamines, along with the other inflammatory mediators also they are going to be released. Inflammatory mediators means, what are the inflammatory mediators? Like a prostaglandins, cytokines, which are responsible for the inflammations. Okay, prostaglandins, cytokines, all they are going to be released. Okay, all inflammatory mediators release when it will be undergoes the process is called as the exocytosis, and this process we are also called as the degranulation. Degranulation means there is the number of cell material, cell contents, it is going to be large cell content they are going to be released from the mast cells. That process is called as the degranulation. This is the mechanism of the histamine. Now we are going to discuss where these receptors are located. I told 4 types of receptors, H1, H2, H3, H4, which are responsible for the various like inflammations, like allergy, like many things. Okay. Then how they are going to be, where they are going to be located, what type of functions they are going to be present. Okay. Then you can see H1 receptors, they are present in the especially smooth muscles. Okay, smooth muscles, what they will be do, they are going to be produce as the allergic symptoms. Allergic symptoms means you might be itching, inflammation, runny nose, sneezing, or different type of things it is going to be occur in our body. Okay, then pain or different things, then that also especially in the smooth muscles. Then what is the effect of this one means, already I told allergy, inflammation, especially of the smooth muscles. The next receptors will be the histamine receptors is the H2 receptors. When the histamine produces its action through the H2 receptors, especially which are located H2 receptors in the gastric parietal cells. Okay, as well as they are also present in the smooth muscles also, cardiac muscles also, immune cells also. Okay, in all the things they are going to be present. Then what they will be do, they are going to stimulate the gastric acid secretion. We might be know acid secretion in the stomach, especially they are going to be produced. Okay, that is the main key function of this one. The next one is the H3 receptors. It is present in the central nervous system, especially in the presynaptic neuron, that is in the brain. what they will be do, it is going to be control the neurotransmitter release. Neurotransmitter release means you might be know the different neurotransmitters like histamine, okay, like dopamine, like norepinephrine, okay, like serotonin, like acetylcholine. All these are the neurotransmitter. It is going to be controlled by the H3 receptors, especially in the brain. By this, what it is going to be happen, we know. The release of the histamine causes allergy. Dopamine you might be take, it is the neurotransmitter. Okay, it is going to be maintain the balance, brain balance in our body, like norepinephrine, and as well as the serotonin you might be know, which is responsible for the happiness and many other things, like the acetylcholine, the movement, it will be control the muscle movement, especially all these body functions it is going to be regulate, especially through the brain by H3 receptors. Then what happen here, especially it is going to be decrease the release of the neurotransmitter release, it is going to be decrease. By that what happened, by this all these functions which is depending on the brain, all the functions they are going to be break down, means they are going to be slow down. Okay, next one is the H4 receptors. These are present in the immune cells. Okay, immune cells means you might be know that one T cells or basophils, or you might be see the other cells, all those cells it will be contain the H4 receptor. Then what they are going to be regulate, this one they are going to be regulate, specially immune cells is nothing but that they are regulate the inflammation and they are regulate the immune response. Okay, it means they never come up, means this whatever the immune response is there, it is not going to be fight with the allergens. That is the reason it is going to be regulate, and it is going to be promote the immune cell migration, means it's nothing but it is going to be promote the inflammation, and as well as the not only inflammation, it is going to promote even allergy also. By what? By controlling the, by regulating the immune cells. Okay, through the H4 receptors. These are all the 4 types of histamine receptors and their location and their functions and their key effects. Okay, next we will be discuss the especially we will be go for the what type of antihistamine drugs are available. Okay, then first we will be discuss about the H1 antagonist. Now we are going to discuss about the histamine antagonists. H1 antagonist also there, H2 antagonist also there, H3 also there, H4 also there. First we will go for the H1 antagonist. Before going to H1 antagonist, we will discuss about what is antihistaminic agents. Antihistaminic agents are nothing but these are the drugs or chemical agents which are used to counteract the action of the histamine. It means what here, which is going to prevent the action of the histamine. The drugs are chemical agents which are going to prevent the action of histamine. Means it might be through H1, it might be through H2, it might be through H3, or it might be through the H4. Now we are going to discuss about the H1 antagonist, means which will be shows the antihistaminic action through the H1 receptors. Here basically they are going to be classified 2 types. One is called as the first generation, another one is called as the second generation. You have to remember the difference between these 2 generations, very important. Here see, the first generation, it is discovered prior to 1960, before 1960. And what is the other difference between these 2? Is nothing but it will be having the high lipid solubility, means high log p value. Log p value is nothing but high lipid solubility. Lipid solubility will be more, means easily cross the blood brain barrier. Okay, it means it will be reach up to the central nervous system. That's why what happened in this case, they are going to produce the sedation. Sedation means it's producing the calmness. Okay, will be how it will be look like this. We are the actionless. Okay, that is called as the sedation. If you are driving the bike, but you don't know the which side the bike will be going, that is called as the sedation effect. Okay, now not only that, it is discovered before 1960. High log p value it will be have, and it produces the sedation. That's why what happens, sedation it is going to be produced means the adverse effects related to central nervous system, means it increases the adverse effects of central nervous system, CNS. What we are discussing here, the difference between the first generation and second generation. First generation, before 1960 they discovered, then they will be having the high log p value is nothing but the high partition coefficient C. Then that's nothing but this will be increases the sedation. Okay, due to high log p value, they are going to be cross the blood brain barrier. It is entering into the blood brain barrier into the CNS, means producing the central nervous system adverse effects. Now come to the second generation. Second generation means the after 1980, whatever the drugs they discovered, after 1980 they introduced this drugs, second generation. Then this will be having the low lipid solubility, means low log p value. Low log p value in the sense what here? They are easily not crossing the blood brain barrier, means no CNS adverse effects. I cannot say no, very less compared to the first generation. The main thing is that the second generation produces the no sedation. What is the difference between the first generation and second generation? The H1 antagonist, basically two types. One is the first generation H1 antagonist, another one is the second generation H1 antagonist. In the first generation H1 antagonist, they discovered before 1960, then they will be have the high log p value, that's why easily cross the blood brain barrier to produce the adverse effect related to the brain. That is the drawback, major drawback. In this case, after 1980 they introduce. Then they will be having the low lipid solubility, means they are not going to cross the blood brain barrier easily. Then other thing is what? That is the reason they will be having the less adverse effects related to the central nervous system. The other thing, main thing is the no sedation. No sedation. That is the difference between the first generation and second generation. We'll go for the chemical classes, the classification based on the chemical structure. These are the amino alkyl ethers, means ether linkage will be there in this particular drugs, which are those diphenhydramine hydrochloride and doxylamine succinate and clemastine fumarate. All these are belongs to the amino alkyl ethers. Okay, next one is the ethylene diamines, means ethyl it will be attached to the two amines like this, NH2, then CH2, CH2, NH2. This linkage will be there. That's why they will be called as the triple enamine. Best example is the triple enamine hydrochloride. Okay, next piperazine. Piperazine means they will be having this structure, piperazine structure. Okay, this structure they will be having piperazine. They are the piperazine derivatives like meclazine, cyclazine, chlorcyclazine and buclazine. The next one is the propylamines. Propylamines means propyl attached to the amine. That class is nothing but the chlorofenoramine maleate and trifluridine hydrochloride. These are the example for the propylamines. Next is the phenothiazines. They will be having the basic structure is the phenothiazine. Phenothiazine means we might be know this is the basic structure of phenothiazine. Okay, they will be, for example for this one, promethazine, trimeprazine and methadiazine. The next one is the cycloheptanes, which will be having the dibenzene structure. Then example for that, cyproheptadines, azatidines. The last one is the imidazoline derivatives, which will be having the structure of the imidazole. Imidazole means which is similar to the histamine. Histamine having the imidazole, and this one also having the imidazole like this. Imidazole structure. Next example is the antazoline. Now comes to the second generation. Already I told the importance of the second generation. No sedation they are going to be produced, and no CNS effects, means side effects will not be there, because they are not going to be cross the blood brain barrier, and because the reason is they having the low lipid solubility. Piperidine means they are having the basic structure, this piperidine structure. Example, loratadine, desloratadine, fexofenadine. Next one is the piperazine. Piperazine already I shown they will be having the basic structure, that is called as the piperazine. That's why they are the piperazine derivatives, like citrazine and levocitrazine. Another thing, this also in the market available, which are we are using for the allergy, like runny nose, like cold, and different things we are going to be use. Citrazine also we are using, levocitrazine. The difference I will show this one between these two in the coming classes. The next one is the alkyl substituted propylamines. Best example is the acrivastine. Then the benzimidazole. Benzimidazole means they will be having the basic structure like this. Benzene plus imidazole. That's why it is called as the benzimidazole. Okay, benzimidazole along with piperidine, both the things are present in this case. Best example is the astimazole. This is the classification of the H1 antagonist. In the next class, we will be discuss one by one class. Means all the classes will be discussed along with the structure and its uses. Okay, mechanism of action, we might be know the histamine it is going to be released. That histamine release only it is going to be prevented. That is the mechanism of action. Means they will be counteract the various histaminic, means it is counteract the histamine, means the release of histamine it is going to be prevented, because that's why they are called as the antihistaminic drug, through which H1 antagonist means these drugs preventing the action of the H1 receptor. Okay, the H1 receptor responsible for what I told, allergies. That in detail we will be discussed in the next class. This is the classification and difference between the first generation, second generation, which will be comes under in the H1 antagonist.